Welcome. My name is Derek Leishman and I'm the Pharma Topic Leader for the ICH E14 S7B Implementation Working Group. I'd like to welcome you to today's sessions, and in particular I'd like to remind you of what we spoke about yesterday and introduce today's agenda to you. I'd also like to remind you that these sessions are being recorded, so if you didn't manage to catch yesterday's session or you would like to revisit the material, these recordings will be available for you to review at your leisure. So let's get started. Yesterday, Dr. Strauss introduced us to the opportunity for the new E14 S7B Q&As. We all appreciate that at their introduction, S7B and E14 were not explicitly linked. E14 suggested a QT interval evaluation quite independent of what the S7B results showed, and that was despite the fact that both documents highlight the need for integration of information in a manner which helps form a totality of evidence. The guideline's been quite successful, however, it's not without some room for improvement. We know ICH S7B studies inform on the safety before first-in-human dosing, but then we've also observed that they're largely ignored afterwards. Clinical assessment relies on human QT interval, but we also know that that is an imperfect biomarker for the proarrhythmic risk that we're studying. Furthermore, prior E14 Q&As do allow for TQT study substitution, but under a narrow range of requirements, and those requirements include the need to study higher levels of drug exposure. In circumstances where a conventional TQT study isn't possible, the clinical evaluation can often be less definitive and result in unclear risk with a no large QT effects label. Yesterday, Dr. Strauss introduced and gave an overview of the new Q&As for ICH E14 and S7B. He was followed by Dr. Garnett, who introduced some example scenarios where the non-clinical data could have an impact on clinical development and interpretation. Dr. Lee then talked about the ICH S7B integrated risk assessment and how this related to those scenarios and some additional scenarios. Finally, Dr. Valentin combined the S7B integrated risk assessment into an integrated non-clinical-clinical risk assessment. The session wrapped up with some discussion of questions received from the audience, and that was facilitated by Dr. Strauss and myself, with all of the speakers and some additional members of the Implementation Working Group. We heard about the value proposition of the new E14 S7B Q&As through directed scenarios where non-clinical data could reduce further the number of standalone QT studies that were required. That would help streamline drug development. We also heard how the non-clinical data could inform clinical regulatory decision-making at the time of a marketing application. That would help inform labelling to better communicate risk, so that no large QT effects label could potentially become one that states low risk of proarrhythmia. The Stage 1 S7B Integrated Risk Assessment Q&A talked in particular of two scenarios to use non-clinical data to inform clinical decision making. We then talked in detail about two particular clinical scenarios which relied on the non-clinical double negative scenario. You'll likely be familiar with how that S7B integrated risk assessment relates to the overall testing strategy that was proposed in this diagram from the original ICH S7B guideline. The ICH S7B Q&As that we heard about yesterday were Q&As 1.1 and 1.2, which covered the integrated risk assessment on how the non-clinical data could be used prior to human testing and later in clinical development for E14 scenarios. In today's talks, we'll be looking forward to some Q&As covering ion channel best practice considerations as well as best practice considerations for in vivo QT assays, and those are Q&As 2.1 and 3.1 through 3.5. We'll also be looking forward to stage 2 in addressing some Q&As concerning best practice for myocyte assays. Those are Q&As 2.2 through 2.5. Human myocytes have emerged as a technology useful in proarrhythmia assessment since the original introduction of ICH E14 back in 2005. We'll also be considering some principles of proarrhythmia models to inform the choice of follow-up studies as suggested in that original S7B testing strategy diagram. So today's order of events are as follows. I'm here recapping day 1 and introducing you to day 2. Next, we'll have Drs. Wu and Jinton who take us through the best practice considerations for in vitro studies. Then Dr. Tsunoda from PMDA will take us through some best practice considerations for in vivo QT studies. And then Dr. Yoshinaga will take us through those principles of proarrhythmia model Q&As. We'll wrap up again with a discussion of questions from the audience, and that will be facilitated by myself and Dr. Strauss. We'll be joined by all of the speakers as well as some additional members of the ICH E14 S7B Implementation Working Group. Before we go further with today's sessions, let me first remind you of what will happen after today. So the Q&As are currently open for public comment. In the United States, that ends on November 30th, 2020, and that's more or less the time it ends in most of the regions around the globe. After then, the Implementation Working Group will be working through the comments and potentially updating the Q&As. Hopefully we'll manage to finalise those Q&As in the middle of 2021. Following finalisation, the Implementation Working Group will work on some training materials which will be released again hopefully in January 2022. Then in 2022, the Implementation Working Group can turn their attention back to the Stage 2 Q&As. It's not just the Implementation Working Group who have work to do, you have work to do as well. We want your feedback, we want your questions today through the Q&A pod, and we also want you to provide official comments during the public consultation periods. Finally, it only remains for me to thank the other members of the Implementation Working Group who've worked to bring the Q&As to the stage that we find them today. And so with that, I'll hand the podium over to Drs. Wu and Jinton. Thank you.
Thank you, Derek. I'm Wendy Wu. Gary, Jinton, and I will be presenting on the S7B best practice considerations for in vitro study. First, let me give you a summary of the S7B testing strategy and the impact of this guideline. This is a figure in the current S7B guideline, and it highlights the non-clinical testing strategy for assessing the risk of delayed ventricular repolarization and QT prolongation. There are 2 core S7B assays. The first is the in vitro hERG or IKR assay. hERG channels are potassium channels thought to mediate the delayed rectifying potassium current, also known as IKR. IKR is important for membrane repolarization in native myocytes. Block of hERG channels or inhibition of IKR can lead to action potential prolongation, therefore QT prolongation. The second S7B core assay is the in vivo QT assessment that is a part of the non-rodent cardiovascular safety pharmacology study. S7B guideline also recommends follow-up studies as needed. And these follow-up studies include the human ventricular myocyte assays to assess repolarization and arrhythmia, multi-cardiac ion channel assays to understand drug effects on the repolarizing and depolarizing cardiac ionic currents, an in silico myocyte model that integrates electrophysiology results to predict TDP risk. Since implementation of S7B in 2005, these in vitro studies have been used to bring investigational new drugs to human studies safely, and they do so by identifying drugs with large effects on ventricular repolarization that could impact clinical trial design. So from this perspective, S7B has worked well for first-in-human studies. Now, experiences tell us that there is still room for improvement. Original S7B provided general principles and no guidance on technical details for the core and follow-up studies. As a result, diverse experimental protocols and practices were used, contributing to a wide degree of data variability. In terms of hERG assay, this would be variability in the IC50 values generated by different labs. For hERG, IC50 is used to calculate safety margin to inform the likelihood of a drug in delaying ventricular repolarization by acutely blocking hERG channels under clinical setting. Large IC50 variability was okay for the data to inform large effects on ventricular repolarization, but data variability makes it difficult to use them later in the clinical development program, as stated in the revised E14 Q&As 5.1 and 6.1. The original S7B also lacked guidance on the non-clinical integrated risk assessment strategy. That means how to interpret the core assay results and when to conduct follow-up studies. And therefore, the S7B Q&A document was created. S7B Q&A 1 was presented yesterday, and it deals with principles of non-clinical integrated risk assessment. S7B Q&A 2 focuses on best practices for in vitro hERG assay and follow-up human myocyte studies. S7B Q&A 3 will be presented in the next presentation, and this covers best practices for in vivo QT studies. This slide summarizes the objectives of S7B Q&A 2. The first objective is to harmonize approaches, reduce data variability, and allow for better translation of non-clinical results to clinical findings. The second objective of S7B Q&A 2 is to incorporate conventional and newer technologies and preparations to assess drug effects on ventricular repolarization. This is an option when there is a positive signal from the core assay to assist TDP risk assessment. Now I will go over the best practice recommendations for the patch clamp studies, which include the in vitro hERG assay performed using overexpression cell lines. This figure shows hERG current recorded from one cell. The current was evoked with the voltage signal below. Now the current is largely abolished by hERG channel blocker E4031. The next three panels on the right show recordings of cardiac L-type calcium current, late sodium current, and peak sodium current, also from overexpression cell lines. The schematic diagram on the right illustrates the contributions of these ionic currents to the ventricular action potential. The original S7B did not provide recommendations in terms of hERG assay design. The new S7B Q&A 2.1 recommends for recordings to occur at physiological temperature, defined as 35 to 37 degrees Celsius. And this recommendation stems from our understanding that effects of some drugs are temperature sensitive, and currently there is no method to predict which molecule would exhibit temperature-dependent effects or the magnitude of these effects. And here are some references that show the dependence of hERG potencies on temperature for some drugs. Another recommendation is that the voltage protocol used to evoke cardiac ionic currents, including hERG current, should incorporate key elements of a ventricular action potential configuration and applied at relevant rates. This recommendation stems from our understanding that drug channel interactions can be state and rate dependent, and here are some references. Next, I will talk about best practices with respect to test article concentrations and exposure. The original S7B guideline states that ascending concentration should be tested until a concentration response curve has been characterized or physical-chemical effects become concentration limiting. The new S7B Q&A 2.1 recommends testing multiple concentrations that cover the steep part of the concentration inhibition graph. This is typically between 20 to 80% current inhibition. The original S7B guideline states that adsorption to glass or plastic or non-specific binding to the test matrix can reduce the concentration of the test substance in the incubation or perfusion medium. The new S7B Q&A 2.1 recommends verifying concentration using solution samples collected from the test apparatus. Solution samples can be collected during the real experiment while recordings take place, or during satellite experiments during which the perfusion apparatus is set up just as in real experiment for sample collection purpose only, but the recording does not take place. And here are some references that show drug loss during electrophysiology experiments and the impact of using verified versus nominal concentrations on drug potency estimation. This slide talks about assay sensitivity and recording quality. The original S7B guideline states that a submaximal concentration of a positive control substance should be used to demonstrate assay sensitivity. The new S7B Q&A 2.1 recommends that for positive control substance, a full concentration inhibition graph should be generated to allow for better estimation of positive control's effect and hence better understanding of the assay sensitivity. Regarding the negative control, time course plots of the ionic current amplitude and other cell health parameters should be constructed to demonstrate stability of the assay under the experimental condition. Later in the presentation, I will show you what these time course plots look like. S7B guideline did not provide recommendations on recording quality. The new S7B Q&A 2.1 recommends that the following be illustrated: adequate control of membrane voltage, manageable levels of background and leak currents, and stability of the recording. The next few slides are on data summary. The S7B guideline did not provide recommendations on data summary in the non-clinical study report. To make comparisons between clinical doses and in vitro concentrations easier, the new S7B Q&A 2.1 recommends providing both molar mass and mass concentration and molar concentration in the non-clinical study report. In table format, report percentage or fractional current inhibition by the test article at each tested concentration for individual cells as well as group average. And this will be mean plus or minus some measures of variability. Here is an example table. The left-hand column shows the drug concentrations tested, and the effects of 13 cells are reported with which drug concentration tested in which cell clearly labeled. The next column shows the total numbers of data points for each drug concentration, and the three columns on the right show group statistics, including the mean fractional inhibition at each drug concentration, SD and SEM. The original S7B guideline states that the duration of exposure should be sufficient to obtain steady-state electrophysiology effects. In graphical format, the new S7B 2.1 recommends that time course plot from individual cells should be included. And the following parameters are to be constructed in terms of time course plots. This will be the ionic current of interest. Here I'm showing you a plot of Herc current amplitude plotted against trace number for the entire duration of the experiment. And you can see that when test substances were applied is clearly labeled. The light gray bars illustrates the number of data points averaged to calculate drug effects for a given cell. The next plot is the input resistance. This measure is estimated using a subthreshold voltage step to elicit a current response and calculated using Ohm's law. Input resistance can be used to inform seal quality and membrane integrity. Now the final plot is the holding current at rest. This plot, along with the input resistance plot, can be used to indicate cell health for the duration of the recording. The figure on the right shows Herc current traces obtained in control solution and following drug application. Note that at the beginning of every trace, there is a small hyperpolarizing response evoked by subthreshold voltage step, and this is used to calculate input resistance for every trace. The black trace is the last trace obtained in control solution. Dark gray trace following 0.1 micromolar application of the drug. Light gray following 1 micromolar application. And finally, red trace is following saturating concentration of Herc channel blocker E4031. Two additional pieces of information are important to include in the non-clinical study report. Where and how important measurements were made. Now, in terms of Herc current, this is typically measured as the amplitude of the tail current. Is this amplitude the absolute amplitude, the absolute peak? Or is it the average value for a current region defined by a pair of cursors? As you can see in the red trace, there is residual outward current in the presence of E4031. So this is not Herc current. This could be background current expressed by the cell or leak current in the presence of E4031. Is Herc current E4031 subtracted or not? This is also an important piece of information to include in the non-clinical study report. The primary endpoint measurements for the Herc assay are IC50 values and the Hill coefficient. The new S7B Q&A 2.1 recommends plotting concentration inhibition relationship using individual data points and group average. This plot is illustrated on the right-hand side. On the y-axis, you see that fractional inhibition of Herc current is plotted, and the x-axis is the drug concentration tested. Individual data points are shown as open red symbols and group mean plus or minus SEM in black. Fit individual data points with Hill equation. Here, the solid red line shows the fit, and the dashed red lines show the 95% confidence interval. Here are the fitting parameters. State constraints placed on the fit. In this case, the minimum and maximum inhibitions were constrained to 0 and 1 respectively. Finally, give measures of variability for IC50 and the Hill coefficient estimates. In this case, IC50 and the Hill coefficient are reported as mean plus or minus 95% confidence interval. This concludes my presentation of the Q&A 2.1. In summary, S7B guidelines should continue to be followed to obtain non-clinical data to support first-in-human studies. The new S7B Q&A 2.1 provides best practice recommendations for patch clamp studies, including the core in vitro Herc assay. The objective here is to harmonize approaches to enhance data reproducibility and allow for better translation of non-clinical study results to clinical findings. The intent is to provide a robust herd safety margin for use in the revised ICH E14 Q&As 5.1 and 6.1. And we would like to encourage applying these best practices early in the development. This could prevent repeating the assays later during clinical development programs. Next, Gary Jintan will present on the best practices for in vitro human myocyte repolarization assays.
Thank you, Wendy. My name is Gary Jintan, and I will be discussing best practices for in vitro human myocyte repolarization assays. These assays are discussed as Q&As 2.2, 2.5, follow-up studies on delayed or altered repolarization. By way of introduction, repolarization represents the integrated cellular response involving multiple overlapping inward or depolarizing currents indicated in red, as well as outward repolarizing currents shown in green on the right-hand panels here. And we're looking at repolarization here with human-induced pluripotent stem cell-derived cardiomyocytes or acutely isolated human ventricular myocytes. There are two electrophysiologic study approaches that are discussed in the Q&As. Both provide recordings of electrical activity which reflect cellular repolarization. The first are transmembrane potential recordings that are obtained using either intracellular recordings with patch or microelectrodes or voltage sensing dyes. The output of these, the measurement derived from these, are the action potential duration or APD. The other approach involves recording the extracellular field potentials that emanate from the myocytes that are above electrodes in multi-electrode array recording platforms. So here we are recording the field potential duration, or FPD, which is measured from the depolarizing waveform to the peak of the repolarizing waveform. Calcium transients and contractility measures may provide surrogate evidence of repolarization changes that are downstream of electrical effects, but they don't represent direct electrical measurements and are not considered further in these Q&As. Some examples of delayed or altered cardiac repolarization are shown in this slide. The first panel shows action potential duration prolongation with the selective blocker E4031 with continued prolongation with escalating concentrations. One can see similar type of effects with prolongation of the field potential duration, here again with E4031 over the same concentration range, prolongation of the repolarization peak waveform. So her current inhibition reduces outward current and elicits delayed repolarization. Excessive inhibition, however, promotes altered or interrupted repolarization, such as early after depolarizations, which is shown in the right-hand panel, which shows this rather aberrant repolarizing waveform that includes a depolarizing spike. This is termed an after depolarization since it occurs after the main depolarizing waveform, and it occurs early or before full repolarization, hence the name early after depolarization, and it is one of the hallmarks of excessive loss or reduction of net outward current. Primary repolarization endpoints from these two measures are indicated in this table here. For action potential duration, as we discussed, it's APD 90, measuring 90%, the time for 90% of terminal repolarization from either single microelectrode or patch electrodes, or from voltage sensing dyes based upon optical field measurements. One also needs to include measures of the incidence of interrupted or altered repolarization, including early after depolarizations. Field potential duration uses analogous measures, but here FPD is measured a little bit differently from APD, right? But still one records repolarization delays. And one can also record altered repolarization, although EADs are a bit more difficult to detect with field potential measures, and they may appear as extra beats. But as with the APD, their incidence as well as characteristics should be reported. The endpoints of these assays can also be described visually as shown here. Here we have a graph illustrating the effects of a drug, increasing drug concentrations across the x-axis 1 through 4, based upon repolarization prolongation in milliseconds. It's represented by the height of the blue bars. And we have, with increasing drug concentrations 1, 2, and 3, greater prolongation. The bars also have error bars indicating the variability associated with the measurement. They also indicate the number of wells that were used to obtain these mean values, the number of wells recorded adjacent to each bar. The right-hand side of the graph illustrates the arrhythmic events, that is the number of wells in which one may see altered or early after depolarizations, as well as the incidence of quiescent events, the number of wells in which case spontaneous beating may cease because of the drug. This is important to understand in terms of the amount of prolongation which is not shown, for example, when 50% of the wells are arrhythmic. And again, one is expected that these measures are baseline corrected, vehicle corrected, and rate corrected. And the data should be provided in tabular as well as graphic formats. In terms of the describing the preparations and experimental conditions, it's important to describe the origin of the preparations, the culture conditions, and the baseline electrophysiological characteristics of the myocytes. So here we're talking about the cell sources, the time and culture, the substrate the cells are grown upon, the media that's used, as well as the technology platform. The criteria for acceptance of the preparations and recordings should be specified, as should the spontaneous beat rate for non-paced preparations. The details of the experimental approaches are also important. The temperature of the preparations, the platform used, the stability of vehicle controls, the time course of drug equilibration, correction factors that are used for the non-paced preparations, the analysis software used, the statistical plan, as well as validation of drug exposures in the testing chambers. It's important to characterize the assay sensitivity for these myocyte preparations. And here we're talking about sensitivity to hERG or IKR current inhibition, which should be demonstrated with non-saturating concentrations of selective blockers such as E431 or tafetalide. And here we're exploring the concentration-dependent prolongation of a positive control with these myocytes. A sensitivity of inhibition of depolarizing in recurrence, the late calcium and late sodium current, should also be provided when multi-channel block is suspected. And here we're characterizing the effects of calcium current inhibitors such as nifedipine or nisoldipine to shorten repolarization, as well as the effects of nixilitine or lidocaine, two inhibitors of late sodium current, to demonstrate repolarization shortening with these agents. The selectivity of the blocking agents used and potential confounding changes in the spontaneous rate, should these occur, should be discussed. So overall, these updated best practices for these in vitro follow-up studies should guide their evolving role in the comprehensive electrophysiological studies of ventricular repolarization, as well as guide the type and amount of information submitted to regulatory authorities for studies with these human-derived cardiomyocytes. In terms of an overall summary of the hERG current Q&As as well as the myocyte Q&As, for the first part, for the ion current measurements, these best practices should harmonize approaches, reduce data variability, and allow for better translation to clinical findings. Especially important is the establishing the validity of the IKR measures in cases where one is supporting the double negative findings from core assays used to support the E14 Q&As 5.1 and 6.1. Add to these advantages the follow-up studies with the myocytes to be able to incorporate conventional as well as newer technologies, as well as human preparations to assess drug effects on repolarization. These are optional to clarify positive signals from the core S7B assays within an integrated risk assessment. And with that, I'll turn the podium over to Dr. Tsunoda, who will be discussing best practice considerations for in vivo QT studies. Dr. Tsunoda.
This presentation is to introducing best practice consideration for in vivo studies. Let me start by introducing myself. I'm Satoshi Tsunoda from PMDA and topic leader in ICH E14 S7B IWG. First slide shows that introduction and background. Since implementation of ICH S7B, in vivo QT studies have been successful as a part of the core battery assay to safely bring investigational drug to human studies. Over the last 15 years, lessons have been learned on how to best perform and report the result of in vivo assay. As there can be some variation in how the studies are performed, the best practice Q&As bring attention to certain considerations. In addition, the new E14 and S7B Q&As indicate that non-clinical assays can contribute to an integrated risk assessment for TDP in later stage of development when clinical data are available. Some additional consideration applied in those scenarios. Next slide shows that summary of in vivo best practice Q&As. In vivo best practice has 5 Q&As that cover consideration for species selection and study design in Q&A 3.1. Exposure assessment in Q&A 3.2. Heart rate correction method in Q&A 3.3. Evaluating assay sensitivity in Q&A 3.4. Presenting the PD and PK results in Q&A 3.5. The aim to these Q&As is that reinforce lessons learned from the past 15 years and how method and result should be communicated to regulators, and highlight additional consideration for assessing drug exposure if the data will be used for the E14 Q&As 5.1 or 6.1. Demonstrating assay sensitivity if the data will be used for the E14 Q&A 6.1. Next slide shows that best practice consideration for species selection and study design in Q&A 3.1. Current S7B guidelines should select and justify the most appropriate non-rodent species. Q&A is recommended preferable to use the same species as non-rodent toxicity studies. Facilitates understanding of potential relationship, cardiovascular, pharmacodynamic effect, and toxicity, and provide complementary information on exposure level. In current in vivo QT study, conscious, freely moving telemetrized animals are used customarily. In addition, alternative model may be justified to achieve adequate exposure to overcome drug-related changes, that is, heart rate change, tolerability, and bioavailability limitation. Next slide shows the best practice for the exposure assessment in Q&A 3.2. Q&As of exposure assessment consists 6 parts. Outline of exposure assessment. Estimate drug exposure level. Best practice of PK sampling from same or separate animal to avoid interference is PD effect. Best practice of choosing relevant time point to utilize exposure response modeling. Example for exposure response modeling by the best practice. And utilize exposure response modeling for human safety. We will explain point of considers in each part by following the slide. Next slide shows considerations for achieving adequate drug exposure. S7B states that drug exposures should include and exceed anticipated therapeutic concentrations. If the data are to use to support clinical decision-making under ICH E14 Q&A 6.1 or 5.1, as exposure should cover maximum anticipated high clinical exposure scenario. Defined as exposure in patients when the maximum therapeutic dose is given with intrinsic or extrinsic factors. Detailed definition of exposure in patients is presented in E40 Q&A by Christine. Next slide shows consideration for assessing drug exposure. Assessing exposure in same animals used for QT assessment is encouraged. When assessing exposure is done in separate animals, separate PK and toxicity study could be used. Blood samples should be taken at relevant time point and in manner that limited interference with QT assessment. Exposure assessment can be done by sampling complete PK profile in same animals on separate day after the adequate washout, or by using limited samples from the QT assessment day to demonstrate consistency with full pharmacokinetic profile generated in different animals in separate study. Next slide shows an example for PK sampling by in vivo best practice. The time point in blood sampling should not be interfere PD assay. Left figure shows in case of using same animal in same study. Right figure shows in case of using the separate animal in same study. Next slide shows a consideration for when to utilize exposure response modeling. If sufficient PK sampling is performed, exposure response modeling similar to concentration QTC analysis for clinical QT studies can be performed. This can be helpful when the non-clinical in vivo QT assay should be powered to detect an effect similar to dedicated QT studies in humans. For example, when using in vivo QT data to support clinical decision-making under the ICH E14 Q&A 6.1. In addition, exposure response modeling may be helpful in other circumstances when QT prolongation is observed or anticipated based on hard assay result. Representative method for non-clinical in vivo concentration QT modeling is published following 2 articles. Next slide shows the best practice for adequate PK sampling to utilize ER analysis. Red arrow shows a representative adequate time point of blood sampling, including high and low drug concentration point in blood. The PK result from adequate sampling time point could be shown sufficient to utilize for the exposure response modeling. Next slide shows a representative exposure response modeling in positive and negative drug. The result of ER modeling of QT positive and negative drugs in left figure are corrected by similar method of the best practice. Red square is shown in QT positive drugs and blue square is a negative drug. The detailed method is shown on the right side. The result indicates that ER modeling could be done and taken the suitable result by the best practice. Next slide shows representative results that ER modeling could be powered to detect an effect similar to dedicated QT studies in humans. Left figure shows ER slope in monkeys administered QT positive drugs were good agreement with humans and shows a slightly similar slopes in appropriate ER modeling. Right table shows predicted delta-delta QTCA effect at Cmax in animals and humans. Non-clinical ER modeling could identify the QT effect consistency with the outcome in humans. Next slide shows that best practice for the heart rate correction method. Independence of QTC to RR interval should be demonstrated through QTC to RR plot accompanied by additional information. Representative independence of QTC to RR shown is demonstrated by the plotting QTC versus RR shown in the left figure. Additional information to demonstrate the independence could be shown. Number of matched QTC RR plots, correlation metric, 95% confidence interval, and p-values. Next slide is how-to choice of heart rate correction when the test drugs affect the heart rate. In the left figure, various heart rate correction method influences MSS slope in intact monkey. These results suggest that correction method for human is not suitable for the animals. Only individual rate correct QT can make the appropriate correction. Therefore, individual QT correction is the best practice when drug affect heart rate. Next slide shows assessing assay sensitivity in vivo QT assay. Test system for an in vivo QT assay should be provide a robust response. As a positive control is not routinely used in the in vivo QT study, assay sensitivity is commonly validated when introducing or changing the test conditions in each laboratory. If study result will support ICH Q&A 6.1, then study should be powered to detect a QTC prolongation effect of a magnitude similar to dedicated clinical QT studies. The best practice consideration provides guidance for assessing assay sensitivity, including when using the historical positive control data. Next slide shows how to demonstrate assay sensitivity. Demonstration of assay sensitivity in each laboratory can be achieved by defining minimum detectable difference of positive control and testing the effect of positive controls. Statistical power calculation could also be provided from the historical positive data from the same laboratory using an identical protocol. If historical positive control data are utilized to justify assay sensitivity, or if statistical power is calculated from the historical positive data, variance of the present data should be consistent with the seen historically. Next slide shows how to presenting pharmacodynamic and pharmacokinetics result of in vivo QT assay. In PD content, summary table and figure is shown. Absolute mean value, mean percent change from baseline, confidence interval, p-value for changes from the baseline, and vehicle control. In PK content, summary statistics for Cmax, AUC, and Tmax for parent drug and metabolite are shown by table. Time plot versus plasma concentration for parent drug and metabolites are shown by figure. Next two slides show a summary in vivo best practice. Best practice in Q&A 3.1 to 3.5 could be applied when conducting current in vivo QT assays. In Q&A 3.1, species selection and study design, conscious freely moving telemetrized non-rodent animal is recommended. To conduct dedicated ER modeling in Q&A 3.2. In Q&A 3.2, exposure assessment, ER modeling may be helpful in circumstances when QT prolongation is observed or anticipated based on HAG assay result. In Q&A 3.3, heart rate collection method, individual rate collect QTC is suitable when drug affects heart rate. In Q&A 3.4, assay sensitivity, best practice for assay sensitivity could be applied when introducing new or changing test conditions in each laboratory. Best practice in Q&A 3.2 and 3.4 should be applied in vivo QT assay when study result will support ICH E14 Q&A 5.1 and 6.1. Exposure assessment in Q&A 3.2, ER modeling can be helpful when non-clinical in vivo QT assay should be powered to detect an effect similar to dedicated QT studies in humans. The representative test conditions for ER modeling are shown in this Q&A. When study result will support ICH E14 Q&A 6.1, assay sensitivity in Q&A 3.4, in vivo QT assay system in each laboratory could be justified assay sensitivity by using positive control or by utilizing historical positive data to define variance and sensitivity. Next slide is references. Detailed test conditions and method could be referred to in these articles. That brings me to the end of my presentation. Thank you for your attention. Next presentation is Principle of Proarrhythmia Model Q&As.
I'm Takashi Yoshinaga, a topic leader of JPMA. I'd like to talk about principles of proarrhythmia models. This is a diagram in current ICH S7B guideline. We discussed integrated risk assessment yesterday. Today, best practice HAG-IKR assay and in vivo QT assay were presented. And best practice myocyte assays was also presented. Last one, principles of proarrhythmia model, is my talk. This slide shows definition of proarrhythmia model. We can use proarrhythmia models such as in silico, in vitro, ex vivo, and in vivo models, which can predict proarrhythmic potential for drug candidate. And proarrhythmia models should follow 6 principles to evaluate whether the model could be used as part of integrated risk assessment. We input some preclinical data into the model and get the output. Regarding model input, we use various pharmacologic data depending on each model. For example, unchanneled data for in silico model, unchanneled data or drug-induced repolarization change, arrhythmia for in vitro model, drug-induced ECG parameter change, arrhythmia for ex vivo or in vivo model. However, model output is similar among these different models. Many models get proarrhythmia metrics. I will show you some examples which have a potential for prediction model in next slide. Example 1 is an in silico model. Unchanneled data is input to model and get net current charge QNet to predict TDP risk as output. Example 2 is an ex vivo canine Purkinje fiber model. Action potential parameter changes, reverse use dependency or others, are used as input and get proarrhythmia score to predict TDP risk as output. Example 3 is an in vitro human iPS cardiomyocyte model. Field potential change, ratio between proarrhythmia and therapeutic concentration are used as input and get two-dimensional map to separate drug into risk categories as output. These examples have the potential as proarrhythmia prediction models. I'd like to say a proarrhythmia prediction model should be follow 6 principles described in the next slide. This slide shows consistency and integrity of general principles. In 2004, a set of principles for the validation of quantitative structure-activity relationship QSAR models for regulatory purpose was established by the Organization for Economic Cooperation and Development, OECD. These principles were adopted by ICH Guideline M7R1 to evaluate the acceptability of QSR models for the regulatory assessment of mutagenic impurities in pharmaceuticals. In 2019, white paper was published with these 6 principles, and in this ICH S7B Q&A, same 6 principles were described. The principles should also apply to proarrhythmia prediction models that are used as part of an integrated risk assessment for regulatory purposes. I will talk about 6 principles from next slide. A defined endpoint consistent with the context of use of the model. For TDP risk prediction, a series of reference drugs with non-clinical TDP risk should be used to define the endpoint. A defined scope and limitations of the model. To generate model input, defined, standardized experimental protocol is necessary. And understand what mechanisms are covered and not covered by the model is needed. A pre-specified analysis plan and criteria to assess model predictability. Training and validation steps should be separated. Before validation, performance metric and acceptable criteria should be specified. Fully disclosed algorithms to translate experimental measurement model input to proarrhythmia risk model output. Enough transparency for independent evaluation of the model is needed to re-evaluate the model performance. Uncertainty quantification. The uncertainty in the model input, experimental measurement should be captured and propagated to the model predictions. A mechanistic interpretation. Need to describe the relationship between model input, experimental measurement, and mechanism of arrhythmia. In this slide, I explain about model development process. According to principles of defined endpoint, clinical risk categorization or quantification should be performed, and 2 compound sets, a training set and validation set, are prepared. Pharmacological assessment should be performed according to these 2 principles. Platform development mechanism investigation should be performed according to principles of defined scope and limitations of the model. Training compound data is input into the platform for model training, and specified model and performance criteria is set after model training, and validation compound data is input into the trained model. Model validation is finished if we could get enough performance for risk prediction. In the next slide, I will explain about model implementation. After a model was developed by the laboratory using a series of reference compounds, the model can be used by other labs. New labs should use a subset of the reference compounds to internally calibrate and validate the model. Example of selecting the subset calibration compounds and performing lab-specific calibration and validation are described in this paper. I will show a diagram of model implementation process. In the model development lab, prediction model was developed using training and validation compound set, as I presented before. In the model implementation lab, calibration compounds are selected. According to these principles, lab-specific model output values are obtained. Predictivity for calibration compounds risk is judged. When answer is yes, lab-specific validation is succeeded, and we are ready to predict the risk of new drug. However, answer is no. More investigation is needed. Here is a hypothetical example when sponsor use the model. A sponsor submits an application for drug A. It is not double negative. It has a positive hug and or in vivo QT signal. Follow-up studies demonstrated that drug A blocks hug and an inward current, such as L-type calcium current or lead sodium current. In the application, the sponsor referenced an ion channel in silicon model previously published or previously went through a qualification process with the regulator and showed that the model development follows all the 6 principles. The sponsor or contract research organization, although not the original model developer, used a subset of reference compounds to internally calculate and validate the model. The sponsor then tested the new drug and the model predicts drug A has low TDP risk. This low TDP risk prediction will be used together with other information to inform subsequent clinical investigation and decision making. Here is point to consider for regulatory submission. Use of the model only under the context of use for which it is developed. Set of control compounds should be tested to assess the consistency between the new data and the historical lab-specific validation data to demonstrate reproducibility of the assay model. Some regulatory authorities require formal qualification of models. When the model is included in a regulatory submission, supporting documentation should be provided. This is summary. A proarrhythmia risk assessment model could be used as follow-up investigation when non-clinical core assays are not negative, a part of the integrated risk assessment. The model should have been qualified under consideration of the general principles presented in the Q&A. The models can be used not only by developer but also by other labs with lab-specific validation data. And that's all for my presentation. But I have one thing. I prepared additional slide with reference information. You can check it after the webinar. Thank you very much.
Well, everyone, that concludes our pre-recorded presentations. We thank you that we've heard a number of questions coming in through the QA pod. So we will now attempt to address as many of those questions we can in the time that remains to us. So I'll be joined to answer those questions by the speakers from today, Dr. Wu, Dr. Jinton, Dr. Sanada, and Dr. Yoshinaga. I'll also be joined by other members of the Implementation Working Group. So this Q&A session will be co-facilitated by myself, Derek Leishman from Pharma, and by David Strauss from FDA. We'll also be joined by Eva Rashid from Swissmedic and by Dr. Yusheng Zhao from TFDA, Chinese Taipei. So without any further ado, we'll kind of launch into the questions. So maybe one of the first things I can do is address a question more generically. We've had a number of questions come in along the lines of, what about Model X? So what about a rabbit wedge model? What about a rabbit Purkinje fiber? What about torsad in vivo? What about other models, potential models that we can use? One of the things that we hope that you would see from the last presentation on the principles of proarrhythmia models is that we have created a Q&A which gives a pathway to being able to take one of those models forward and use it as a proarrhythmia model with some recommendations for qualification, etc. So we will be attempting to address the questions that are specific to the other presentations, but those that are more broadly generic to the proarrhythmia models, and is this model acceptable or not, I would refer those questioners to the principles of ProA and to some of the discussion on qualification and positive controls of the other models, so that there's some guidance there. So with that generic piece of information, I'd now like to launch into some of the questions that we have had. One of the big question areas that we've had concerns how to ensure the sensitivity and I guess sensitivity is probably the best way to summarize it, but how to summarize the sensitivity and needs of the in vivo model. So maybe that's a question I could offer to Dr. Tsunoda. So Dr. Tsunoda, we had a number of questions regarding the number of milliseconds in the animals and the relative sensitivity that we need for the in vivo model. Maybe you could address that generically. Maybe I can answer. I could ask the question again, just in case there's some audio issues for Dr. Tsunoda. I'm looking to see if he's in the room. Yep. So Dr. Tsunoda, what kind of in vivo study is recommended when the non-clinical in vivo QT assay should be powered to detect an effect similar to dedicated QT studies in humans? Dr. Tsunoda, you're very quiet. We're not hearing your response. I can hear you very vaguely in the background. Perhaps while we're solving those audio issues, we could move across to a related question on exposure response. So maybe this is a question for Dr. Chiba. So Dr. Chiba, what are the methods that people might consider when they're considering exposure response modeling in the in vivo study?
Can you hear me?
Yes, we can hear you.
Yeah, as for in vivo exposure response modeling, the detailed methods are not described in today's presentation. The representative exposure response model is shown in two references in this presentation, and the summary of the method shown on slide. The key point states in today's question and answer 3.1, 3.2, and 3.3. Adequate enough ECG monitoring and blood sampling for the exposure response modeling could be done by using conscious, freely moving, telemetrized, and animal models. And assessing exposure and ECG monitoring in the same animal is recommended. And there are more time points for compound, such as hysteresis type profile and accumulation of heart. And blood sampling should be taken at relevant time points and in a manner that limits interference with QT assessment itself. In addition, individual rate corrected QT is suitable when a drug affects heart rate. That's all.
Thank you for that. Maybe we can move our questions around a little and make sure that we cover as many as we can. So moving to some in vitro questions. So Dr. Wu, there were some questions on the positive controls for the in vitro assays and does the sponsor have to run the calibration compounds each time? If not, how often? And then there were other questions on how you choose the positive controls. Could you answer some of these questions around positive controls?
Sure, thank you for the question. So regarding the first part, does a sponsor need to run calibration controls each time a new test article is examined? The answer to this question is actually not stated in the ICH S7B Q&A. And here are some thoughts. One practical approach can be periodic generation of the reference data to demonstrate assay reproducibility and sensitivity. Now, this is stated in the S7B Q&A 1.2. The herd safety margin would be established using a set of reference compounds, right? So within this set of reference compounds, then a few can be chosen and tested with new test article. So with time, this kind of data set would provide evidence of assay reproducibility, and then the information can be used to redefine the frequencies of how often the reference compounds would have to be tested in the future and whether the numbers of reference compounds can be adjusted in the future. So that's the first set of questions. The first question. And the second question has to do with the choice of reference compounds. Now, the Q&A is not prescriptive. So the sponsor is welcome to propose a list of reference compounds. And, you know, there are some lists out there, the SIPA 28, JIXA compounds, credible mets, and provide justification in the choice of these reference compounds for safety margin.
Thank you, Dr. Wu. Just there was also some follow-up questions regarding positive controls in vitro assays, regarding which positive controls and how you choose positive controls for the cardiac myocyte assay. That's a question, I think, for Dr. Ginton.
So thanks for the question. Regarding positive controls on the myocyte, it's essential to demonstrate sensitivity of the biology on your study, the myocytes, with positive controls, especially in cases where one is trying to demonstrate that you do not have a test compound that, for example, blocks hERG. It's important to know that the biology that you're studying does indeed express functional hERG current, and this is the importance of using a positive test compound such as hERG, E4 or P1 hERG defibrilate.
Okay, thanks, Gary. Maybe sticking with the in vitro theme, but changing the topic a little. We had a number of questions that came in regarding concentration verification. So perhaps we could start with Wendy, and Wendy, you could give us some of your thoughts on the need for concentration verification in the hERG assay in particular.
Sure. So concentration verification is a part of being GLP compliant, and it's really important, I think, when, especially if a study is reporting negative, one would not, it will be important to know that drug loss has not occurred in the electrophysiology rig to provide that negative result. And even for positive studies, having, determined the IC50 using verified concentrations also gives confidence in applying the IC50 to calculate the safety margin and using that safety margin for decision making.
Thanks, Wendy. And maybe expanding on the question a little and bringing in Gary. So there were some questions on how you would do the concentration verification. So maybe I'll pose that question to Gary, and Gary, if you could kind of answer with respect to both the hERG assay and the myocyte assay, that would be useful.
Yeah, so following upon what Wendy Wu just said, it's important to characterize the drug exposure during the cardiac repolarization and the myocyte repolarization studies as it is with the hERG studies, especially, for example, in cases where there may be no current inhibition, so you're demonstrating sensitivity. It enables better translation of in vitro to in vivo, as well as clinical findings. And the recommendation follows general GLP practices. In terms of how to sample, it's best to sample from the actual test chamber, as there may be drug losses in different parts of the apparatus. For example, if you're using a flow-through system, you may lose drug through the reservoir or through the tubing that leads to the test chamber. So it's ideal to measure the concentration in the test chamber or in the effluent if it's a flow-through system. If it's a non-flow-through system like a well, it could be done using what you might term a satellite well, in which you duplicate the experimental conditions that you had with the myocytes or with the hERG transfected cells, but just do it without those cells present.
Thanks, Gary. Now we'll try and return to the in vivo questions. So, Dr. Satoshi, there was a question for you earlier, which was, what kind of in vivo study is recommended when the non-clinical in vivo QT assay should be powered to detect an effect similar to a dedicated QT study in humans? I'm sorry. It seems that the audio issues with for Dr. Sonoda persist. Maybe I could offer the same question to Dr. Chiba.
Yeah. I described that previously, and the importance of in vivo exposure response modeling. This model can be helpful. I shown the difference at the Komatsu, and also showed the exposure response modeling could be performed with the same number of animals as a current and the in vivo standard QT assay. The number of animals is unlikely to increase significantly when conducting in vivo exposure modeling. And the ER modeling with sufficient power can be used for the specific case, such as question 5.1 and 6.2. And this is also helpful in other circumstances when QT prolongation is observed or anticipated based on hand results. That's all.
Thank you for that answer, and thank you for addressing directly the topic of number of animals, because that was something that featured in a number of the questions both yesterday and today. Maybe I could offer an answer to one of the questions that has come up regarding the absolute number of milliseconds or percentage change in that in vivo study. People were asking a question, what is that number? That number obviously isn't given in the Q&As, which is the reason that the question's coming up. It was not given deliberately because we actually think that there are a number of ways to address that question. One of the ways that you might address that question is to think about for a compound in humans, if it gives 10 milliseconds at a particular concentration, you may be considering, well, how many milliseconds does that same concentration give in animals, and that may be the power that you'd like to drive the animal study to. Equally, it could be a case of you can comfortably measure 10 milliseconds in animals, and so the question then becomes, for the same compound, at what multiple of the human exposure can you detect that 10 millisecond change in animals? So there's a few different ways to address the question, and I've only just given two kind of possible ways to address it. I think that there's some more scope for everyone to address that question in slightly different ways. So I think that that's the reason that answer is not very detailed in the Q&A, because it gives the flexibility to the investigator to develop and justify the metric that they use in that in vivo study. So I'm getting good signals that we may have Dr. Sonoda online and with audio that works. So maybe I can pose the next question to Dr. Sonoda. So Dr. Sonoda, there was a question posed on the gender in the in vivo studies, and do both male and females have to be tested?
Okay. And David, and could you hear me?
We can hear you very well.
Okay, I can connect. I'm sorry. Okay, I'd like to the next and the question is and the provider answer. Please moment. Please wait. Okay, I think is and the gender is and routinely in use for the male, but only used for drug in male, or the metabolize difference and male and difference and between male and female, and this in this case is used for, could be recommended to use for the and female animals. Okay, thank you.
And I'm really glad that we have the audio issues straightened out. So my next question is on another topic that popped up several times in the questions, and it's on GLP. So one of the questions is which assays need to be conducted to GLP? Eva, maybe that's a question I could pose to you.
Yes, sure. Well, first of all, there were several questions I've seen about the follow-up studies, like the myocyte studies and so on. And I would point out that according to the S7B guideline, it is not a requirement to conduct these follow-up studies under GLP. However, they should be conducted under high-quality controlled conditions and whenever possible in a compliance of GLP. With regard to the core assays, we'll say the HERG assay and the in vivo QT assay, these studies should be conducted in compliance of GLP when they are performed for a regulatory submission, as already stated in the S7B guideline.
Thanks, Eva. I think that clarifies for a number of people who had questions on this topic. If we can now move back to some more generic questions on the proarrhythmia models. We did have a question, and I think that there's an opportunity to provide some clarification regarding the question of inputs and outputs. So we had some questions, and I think this is a question for Takashi. So the inputs seem rather obvious when you're talking about an in silico model on the for proarrhythmia. It gets a little bit more difficult when you're thinking perhaps about the in vivo models and the other in vitro models. So you could maybe provide some clarification on inputs and outputs and proarrhythmia models, because I guess there was a feeling from some of the questions that they were very they could see very clearly how it applied to in silico models. I think that there was people a bit more struggling with the in vitro and in vivo models.
Okay. A SIPA model is very famous, so many audience can make it easy. However, as in silico model, ex vivo or in vivo models, we can use some parameter changes, such as some parameters of action potential or ECG parameters, and additionally other index, such as concentration. For example, the margin between therapeutic concentration in clinical and assay concentration. Using these indexes, we could get predicted TDP risk category for new drug candidate. I showed some publications in my slide. Please, but this is just a possibility of the example. So please check it later.
Thanks, Taka. Moving on to another related topic on those proarrhythmia models. The proarrhythmia models, one of the questions that's come up previously and during the webinar here is how you choose the compounds to kind of validate the model if you're using someone else's model. So I guess that's a calibration for model implementation question. So the calibration drugs, how do you select the calibration drugs to perform a lab-specific calibration and validation?
Okay. Regarding compound selection for the calibration and validation, for the implementation of a qualified model in another lab, a smaller subset of the compound that were originally used to train and validate the model can be selected. And in my presentation, I have referenced one recent publication to illustrate how to select these calibration drugs for lab-specific model implementation. So please check it to understand more exactly. Thank you.
Thank you, Taka. So returning to the in vitro theme, we've had a number of questions there. So one of the questions that came up was asking for some further clarification around the rate and frequency questions for the voltage clamp protocol. So this is a question for Wendy.
Sure. So the question that came in was, a pacing rate would be constrained by the duration of the step ramp protocol, for example, that's used to study Hertz current, and also by channel properties, channel gating. And is there additional scenarios and best practices that one would need to follow to assess rate effects on channel of interest? Now, Q&A 2.1 also has these provided guidance on some of the features that will provide confidence in the electrophysiology data, and these are not limited to rates. In terms of stability, the quality of the recordings, the use of positive control to demonstrate assay sensitivity, these all apply. And in terms of experience, yes, we have come across scenarios where additional rates are important to understand patient-specific risks, but these are on a case-by-case basis if they're needed.
Thanks, Wendy. And expanding on the theme of some of those experimental details, we also had a number of questions around the recording at physiological temperature. I think a lot of people would appreciate that can be challenging. Can you maybe expand on the use of physiological temperature and the rationale behind that?
Sure. So this was covered in my presentation. Physiological temperature dependence of drug ion channel interaction is not just a thing that's seen with Hertz channels, it's seen with other channels as well. And so the S7B Q&A 2.1 recommends physiological temperature recording for the relevant ionic currents. Because indeed, so I'll go back to Hertz. Indeed, there have been a number of studies demonstrating that temperature-dependent effects can be expected for some drugs, and right now there is no method to predict which drug is going to exhibit temperature sensitivity or even how big this magnitude is going to be, hence this recommendation of recording at physiological temperature. I think there have been a number of questions coming in regarding, concerns about recording at 37 degrees for sodium and calcium currents, because these can be very fast signal, and relating to voltage control. And I think this can be addressed by a simple experimental procedure where the IV relationship is established prior to the pharmacology experiment. And what one would be looking for here is a graded increase in the current amplitude as the voltage step is increased. And that provides some confidence that one still has voltage control. Relating to calcium current, there is a question that comes in that talks about rundown. The thing with calcium current rundown is that it occurs both at physiological temperature and room temperature, and S7B Q&A 2.1 recommends having long enough control recording to establish steady state in the control level, in the control solution prior to drug application. And I hope that addresses the question.
Thanks, Wendy, and thanks for exploring some of the further questions that have come through in the Q&A. So, returning now to in vivo, this is perhaps quite a detailed question, but there's a question that's come up, and I think it's for Katsuyoshi, on how many animals do you need for the PK sampling? Is there a recommendation for the concentration QTC?
Yes. And in general, in safety pharmacology CV assessment, we use 4 animals per dose used, but for precise and concentration QT analysis, and it's better to large number of animals for improving assay sensitivity and statistical power. However, its important thing is to confirm the assay sensitivity of each facility. So number of animals, 4 animals or more animals is preferred for this CV assessment. That's all.
Thank you. Thank you for the clarification. We've had other questions come in. We're kind of bouncing back to the in vitro theme. So Wendy or Gary, there have been a number of questions concerning whether or not the patch clamp studies are restricted to manual patch clamp or to automated systems. I think that's that was a question to Wendy or Gary, and it's whether or not there is any restriction on the type of patch clamp equipment, if you like.
Sure, I will address that question. The Q&A is not written with a specific platform in mind, so it has, describes data quality consideration and criteria. If automated patch clamp system generated data satisfy all the criteria, then I don't see why they wouldn't be able to be used.
Thank you. So we've had a number of other questions come in on the in vivo study, and one of the questions that came up actually has popped up a couple of times is, and then maybe it relates to both in vitro and in vivo, but let's start with in vivo. And for Dr. Sonoda, the question of metabolites and studying of metabolites, this is something that that's touched on in the Q&As, but people were looking for a little bit more clarification on which metabolites might need to be considered in these studies.
I think it's a very it's a very difficult question, and I think it's a Derek, I'm sorry, and the question is the please again. Okay.
So the question was around are there any general guidelines on the the metabolites which should be addressed?
Yeah, I think it's and human specific and metabolites is observed, and I think is and in vivo QT assay is focused on the human specific and metabolite, and in sometimes is the synthesize and metabolite using a metabolite, and in vivo QT study could be conducted. That's all.
Okay. So I'm returning to the a theme that we touched on earlier, and it comes back to the question on the let's see, I've lost it in the track. Sorry, my audio's just gone out too. Can people hear me? So this was another question for you, Dr. Sonoda. So regarding the in vivo study, is a single time point assessment in during the recording phase plus a full profile from the tox studies, is that a technique that can be used for concentration QTC assessment?
routinely and in vivo QT assay is useful for the toxicokinetic assessment and data. But in the and ER modeling is I recommend the PK profiling in the same study. Okay, that's all.
Okay, thank you. So I guess this is a question. This one's coming back to Wendy. So, Wendy, if a HERG study has been done at room temperature and was negative, and there's no in vivo QTC at high doses, would the HERG need to be repeated at 37 degrees?
So it depends on what this HERG data is being used for. If it's being used to pursue the path described by Dr. Christine Garnett yesterday, the revised E14 5.1 and 6.1, we would recommend HERG obtained under best practice, which would be 37 degrees.
Thanks, Wendy. I think that probably also addresses a number of other questions, and maybe you'd like to expand a little. People were asking about situations where they've already conducted their HERG assay, but they haven't gotten to the stage in development where they've been thinking about questions 5.1 and 6.1. Under those circumstances, would a repeat HERG assay be necessary?
Sure. So again, I think Dr. Christine Garnett was pretty clear to for the HERG data to be used for 5.1 and 6.1. Best practice HERG would be expected, and that will be following the recommendations in S7B Q&A 2.1.
Thank you. So there's a question here, and this one I will pose to Dr. Chiba, and it relates to the in vivo study and its formulation verification. I guess people are just trying to clarify, given the requirements for concentration verification in vitro, they're just asking about the formulation analysis for the in vivo study. So is formulation analysis sufficient to show the animals were correctly dosed?
Yes, thank you for question. And at first, demonstration of independence of QTC to heart rate RR intervals is needed, and QTC and RR plot accompanied by additional information and analysis of QT-RR interval relationships are important. So we requested in almost all cases, individual QT correlation is preferred method, as it is more accurate and sensitive than the general method, such as Bazet, Friedrichia, and so on. In the cases, so when the test drugs affect heart rate, because non-rodent, in addition to because non-rodents show species specific, so individual differences in QT-RR relationship is very important. That's all.
Thank you, and thank you for the answer to which was to the next question I was going to pose to you, which was around heart rate correction. Can we return to the a question on dose formulation analysis for the in vivo study, Dr. Chiba? So is dose formulation analysis sufficient to verify that an animal was dosed correctly? I guess it becomes a moot question if you're doing exposure QTC.
Formulation analysis of course, it's very important analytical analysis. And for the precise and concentration QT analysis and formulation analysis is and mandatory for each time point. Yes.
Okay, thank you for that answer. So it looks like a number of the questions that we've had coming in are relatively similar to ones that we've already addressed. So I think we're starting to see that the questions are drying up. Here's one question that is that picks on something that we touched on yesterday, and this is a question that's coming for Dr. Wu. So for calculating the free Cmax from in relation to Herg margin, if a compound is 99.5% bound, do we use 1% free instead of 0.5%? So that's a question for Wendy.
Yes, this question was addressed by Zhihua yesterday. Per FDA guidance, 1% would be used.
Thanks, Wendy, for that clarification. I'm just quickly scanning through the questions. So Wendy, this is a follow-up question since we're asking you. So this is regarding the temperature of recording, and I guess the question is, would you expect the Herg IC50 value to shift left for physiological temperature compared to room temperature?
Right. So again, Herg IC50 value is a static calculation, right? We're looking at steady state drug binding to the channel, and so if the temperature has different effects on the on and off rates, it could shift the IC50s differently. We have seen both ways, that going to 37 degrees increase IC50 for some drugs, decrease IC50s for some drugs. We can't predict the direction of movement.
Okay, thanks, Wendy, and thanks for reflecting on your experience with drugs moving both ways. So we've still got a few questions coming in, so I am encouraging the audience, if you do have questions, keep them coming. It gets a little bit trickier to sift through them and to point them at specific people to answer them when they're coming in, but this kind of live situation. But we've got time, and we will keep answering as many questions as we can. So there's one question regarding heart rate. So we touched on heart rate correction there. We also touch in the in vivo question on situations where heart rate changes. Is there a cutoff in those in the in vivo studies? So this is a question for Dr. Sonoda, Dr. Chiba on heart rate. And is there a cutoff in terms of heart rate change that makes it difficult to use the conscious animal, and where other models need to be considered? So I guess let me rephrase that question just to make sure that I was clear. So when a drug does affect heart rate, is there a heart rate cutoff where you would think that the conscious animal may not be appropriate, or is this something that is justified on a case-by-case by the investigator?
Yeah, I think the case-by-case approach is very important for in case of the heart rate changes. As previously mentioned, the individual heart rate correction is very important. And also in the very large heart rate changes, it is one of the options of the other alternative and method, such as anesthetized animals and the controlled heart rate. It is very important and one of the options, I think. That's all.
Thank you. So just to make sure that we've covered some of the topics that popped up from yesterday, I wonder if I could ask Dr. Strauss to maybe address some of the questions that popped up relative to the integrated risk assessment and the proarrhythmia models.
Thank you, Derek. Yeah, we had different questions that came in about the use of additional in vitro or ex vivo models, and also about the SIPA in silico model, and how are these applied in the current Q&As. And in the last section of the S7B Q&A 1 on integrated risk assessment, and the first question there, 1.1, we have a section on what to do when the HERG assay and/or in vivo QT assay suggest an effect at clinical exposures. And the Q&A response comments on how under this scenario the drug's torsade risk could be affected by other factors such as other ion channels, effects of trafficking of ion channels or metabolites, and follow-up studies can be performed to further explore the mechanisms and assess torsade risk. And if applicable, the best practice considerations should be followed to the extent possible for the additional ion channel studies or in vitro cardiomyocyte assays. And then it's important to note that an appropriately qualified proarrhythmia risk prediction model that can be in silico, in vitro, ex vivo, in vivo, can be used. And you heard further about the proarrhythmia models considerations today in the last presentation. And a model can then be used according to its context of use. And the assessment of TDP risk, torsade risk, using these follow-up studies, while it's optional, it can be used together with other relevant non-clinical and clinical information to contribute to the design of subsequent clinical investigations and interpretation of their results. So the various assays put forward under the SIPA initiative or other proarrhythmia models can be used within the current draft Q&As. Their exact impact on clinical decision-making is not explicitly spelled out in the current Q&As, and so the cases would be evaluated on a case-by-case basis. And as we discussed and presented yesterday in my initial presentation, the details of how we would propose to use proarrhythmia models in combination with ECG biomarker data to inform clinical decision-making, such as the need for ECG monitoring in phase 3, and regulatory decision-making and labeling at the time of a marketing application, is something that the group plans, the ICH E14 S7B Working Group plans to lay out in more detail in stage 2 of Q&As that would follow finalizing these initial stage 1 Q&As.
Thanks, David. That was great. So we've had a number of other questions coming in, and I think we've touched on the answers to some of these, but given that there's questions still coming in, we'll revisit some of them just to make sure. So, Chiba-san, this was a question that probably relates to an answer you gave, and this is how many animals are generally required for the concentration QTC analysis in the in vivo study?
Yeah, I think the 4 animals is enough for concentration QT analysis described today's presentation. But I think the For more precise concentration QT analysis, of course, large animal number is preferred for improving assay sensitivity and improving statistical power. So I think that it is case by case. It depends on the assay sensitivity of each facility.
Thank you. I think that provides some extra clarity to the answer that we gave earlier. This is a question for Wendy, and this is regarding the report figures. So is the expectation that the current time and the input resistance time and the holding current time plots be presented for each cell, or will representative cells be considered sufficient?
Thank you for the question. This is an important question, and yes, it is expected or recommended for each cell. So this, let's look at what these plots show us. These plots show us that indeed the control recording has reached steady state level and that drug effects are at steady state inhibition. That's your signal of interest, the first plot that I showed. In terms of input resistance and holding current, these are plots of passive membrane properties, and they really speak to the quality of the cell that one is reporting. So together, this package will provide confidence that all of the data points passes through acceptable criteria.
Thanks, Wendy. That's great. That really it will be helpful to people as they prepare these reports. I think we're getting to the stage where a lot of the questions we've covered now. I'm just looking at some of the newer questions just in case there's something that we haven't quite covered. And I'm not really seeing anything that isn't something that's we've already touched on. So given that, I think we're getting to the stage now where we can wrap up today's proceedings, and I'd like to thank the audience for their participation and for all the questions that they have sent. If questions still trickle in, we will of course be keeping all of these questions, and they can roll over into the next phase of the next phase of the proceedings, which is the open consultation period, and everyone's obviously free to give responses and comments and further questions for those open Q&As, and the Implementation Working Group will be receiving those, and we'll be able to address those later in the year and in the beginning of 2021. So with that, I just need to thank all of my fellow participants from the Implementation Working Group and the wider Implementation Working Group who have been working in the background here on some of the questions, and who obviously put in all the work on getting us to the place we are in today. So at this point, I think I can hand back to the organisers to help close out this meeting for us.